Yeah, hi guys. This is my YouTube channel, Zugi YouTube channel, and today I'm going to teach you about biology, biology movement, and I'm going to teach about movement of substances into and out of the cell. If today is your first time watching my channel, please subscribe and like my YouTube channel. Thank you. Okay, so from the starting, like this is the introduction of movements of substances into and out of a cell. So this is the introduction. Cells need substances within to perform the various life processes. These life processes generate toxic substances that would have to move out of the cell to maintain a steady state internally. So all that is trying to say is that since cell is the basic unit of life, that's where everything begins at, and that when a cell goes through like the processes, like life processes, sometimes it takes out substances and sometimes it's taking substances in order for it to become healthy or something like that. Yeah, so in this lesson we are going to look at the need of for substances to move in and out of a cell and the psychological processes that are involved. So objectives, yeah. So by the end of this lesson, you'll be able to explain the need for movement of substances across the cell surface membrane. Explain the concepts of diffusion, osmosis, and plasmolysis, and also importance of movement of substances in and out of a cell. So this is a cell, and this is like when any substance like has to go out of the cell, like because the cell is has a semi-permeable membrane. That means that substances can go in and get out of the substance. So when the cell needs anything, like this part of the cell, like it goes through these processes, the osmosis processes and all those things to in order for like the substances in the cell to move in and out of it, so that the cell will be in a healthy state. So as you continue reading, like living cells of an organism go through various metabolic reactions in which certain substances outside the cells are needed. These substances are transported across the cell membrane into the cytoplasm by various mechanisms. Wastes generated in a cell would have to move out. These transports of substances in and out of cells are important for a number of reasons, and they include so this is like all that trying to say is that like these are the reasons why the substances like need to move in and out of the cell. And some of the reasons why substances need to move in and out of the cell is that for movement of nutrients needed by the cell for energy production and other metabolic processes. The reason why substances moving out of the cells is for like the removal of waste substances produced by the cell during its metabolic activities, and also like the secretion of useful substances such as lipids and proteins, and also the generation of ionic gradient between the cell and its environment for nervous and muscular activities. So yeah, and also the maintenance of suitable pH and ionic concentration within the cell for enzyme activities. And also this is another topic, this subtopic that we have to discuss, and this the psychological processes by which substances move in and out of the cell. So some of the examples are like this: diffusion, osmosis, active transport, and endocytosis, exocytosis, and all those things. So the meaning of so we talk about diffusion. Diffusion is one of the ways by which substances move in and out of the cell. So let's go. Diffusion can be defined as the movement of molecules or substances or particles from the region of higher concentration to the region of lower concentration until the molecule or substance or particle are evenly distributed. Okay, so for distribution to occur, for distribution to diffusion to occur, a concentration gradient must exist between the two regions that are involved in the process. Concentration gradient is a difference in concentration between two regions. So as you can see, like all this is trying to say is that like diffusion is the process is part of the process by which substances move in and out of the cell. So like when diffusion is about to occur in a cell, it has to show like it has to have two sides. One part has to be a place whereby like there is a high concentration gradient, and the other place has to be a low concentration gradient or no concentration, whereby the high concentration has to move to the low concentrated part in order for all the molecules to be evenly distributed in the cell. So that is all I was trying to say. So like an example is, as you can see at the side over here, diffusion, high concentration to lower concentration, so they are even distributed. So that's it. So yeah, on another subtopic, which is experiment to demonstrate diffusion. So like what is this means is that like I'm trying to demonstrate like an example of diffusion like in a substance. So like this is it. In this experiment, a clean beaker is filled with distilled water. A crystal of potassium texomagnate 7 is carefully placed into the water in the beaker. The setup is then placed on a flat surface. The setup is observed for about 30 minutes. After about 5 minutes of the crystal, after about 5 minutes, particles of the crystal would be seen moving from the bottom throughout the water. This continuously until the water becomes totally filled with the particles, and it becomes purple in color. So what this is trying to say is like there are some experiments to demonstrate diffusion. So you put there another name for this thing is that the texture magnet 7 is that like it is called blue, and that's what you put inside like into water and soak white dresses inside. So that is it. So as you can see, when you put the blue inside the bucket, you see that from the bottom it starts to like rise up. When you leave it for a few minutes, you can see that the blue starts to get evenly distributed. So as you can see, that is an example of diffusion. So as you know, so that's it. So let's into factors that affect the rate of diffusion. So yeah, these are the factors that affect the rate of diffusion, which are concentration gradient. The rate of diffusion is is faster with steeper concentration gradient. It is an advantage for a cell to maintain a steep diffusion gradient with rapid transport. So yeah, so concentration gradient is part of like the factors when dealing with diffusion. So and also temperature to its part. So how high or low the temperature would determine how fast or slow the diffusion will occur or something like that. Yeah. So as you can see, this is it. So and the size of the molecules. So this is it. Size of molecules. If the size of the molecules are smaller, that means that it will be able to diffuse faster than when it is and than when it is bigger. So that's vice versa. So it's vice versa. So and also another thing factor that affect diffusion is also the surface area of membrane through which diffusion takes place. The greater the surface area of the membrane, the greater the rate of diffusion. So that's it. So that means if the surface area of where the only permeable membrane where it is passing, if the part where it is passing is greater, that means diffusion will be able to occur faster and vice versa. Examples of diffusion in plants and animal, which is like as you all know, oxygen diffuses out of leaves and carbon dioxide into the leaves through the stomatal pores during the daytime. Yeah, so this is an example of diffusion. Plants Plants absorb some mineral salts by diffusion from the cell through the hair roots. Yeah, example of diffusion. And also oxygen diffuses out of the blood and carbon dioxide into the blood in the tissues of mammals. So yeah, that's examples of diffusion in animals and distant plants. So this is another factor which also helps the cells in the body to be able to move substances in and out of the body, and that is osmosis. So osmosis can be defined as the movement of water molecules across a semi-permeable membranes from a dilute solution to bracket higher water molecule concentration to a lower concentrated solution into brackets lower molecular concentration. And also osmosis can be considered as a kind of diffusion in which only water molecules move. Yeah, so as you can see, osmosis too, it has some similar properties as diffusion, but and there are similar properties that osmosis is mainly talking about water. It doesn't talk about gases and all those fluid. It is only water that osmosis deals with, which is like moving water molecules moving from lower to higher and all those things. So examples, experiment to demonstrate osmosis. In this experiment, a visiting tube of about 15 centimeter long is half filled with 2% sugar solution. With the support of a clamp and a resource stand, lower the viscous tube into a beaker of distilled water. The setup is left to stand for about 20 to 30 minutes. So as you can see, this is some example of osmosis occurring whereby the sugar solution over here has a lower concentration and this place has a higher concentration. So due to that, the water the water move from the other part to this part in order for this place to become stable. Yeah, that's it. As you can see, and factors that so these are the factors that affect the rate of osmosis, and these are the concentration gradient, which is the steeper the concentration gradient, the higher the rate of osmosis, and also temperature. So the higher the temperature, the faster the osmosis rate. So that is it, and vice versa. And permeability of membranes. So as you can see, like how hard or soft the permeability of the membranes will determine how fast osmosis will occur. For example, if the density of the permeable membrane is very high, then that means that osmosis will take a very long time before it will happen. But if the density of the substance is of the semi-permeable membrane is so low, that means osmosis can pass through easily and osmosis can occur. So another topic too that we'll talk about it too is and this thing diffusion and osmosis is that this thing tonicity of solutions, which tonicity of solution is just the measure of the amount of solute concentration in a solution. There are three types of tonicity, which is hypertonic, hypotonic, and isotonic solutions. So hypertonic solutions, this is a substance whose solute concentration is higher than the one it is being compared with. So as you know, so this is a hypotonic solution. This is a solution whose solute concentration is lower than the one is being compared with. So yeah, this are some demonstrations over here: hypertonic solution, hypotonic solution, and isotonic solution. So that's the and isotonic solution. This is a solution whose solute concentration is the same as the one it is being compared with. Okay, so we are done with that. So we are on plant cell in hypertonic or hypotonic solution. So if a plant cell, for example onion epidermal cell, is placed in hypotonic solution, water molecules will move across the cell wall and cell membrane through the cytoplasm into the vacuole of the cell by osmosis. This is because the solute concentration of the cell sap is higher than of the surrounding solution. The cell swells, pushing the nucleus across to the cell walls. However, due to the thick and rigid cellulose cell wall, the cell will not burst. The movement of water builds up pressure against the cell wall. This this pressure is called turbo pressure. So the cell the cell wall produces a counter force called cell pressure. When the turgid pressure becomes equal to the pressure of cell wall, the cell wall is said to be turgid. At this point, the tendency of water to enter the cell is exactly balanced by the pressure potential. This is the amount of water entering the cell is equal to the amount of water leaving the cell. Hence, the net take up of water in and out of the cell is in equilibrium. So all that this is just saying is that, like, when you put an onion epidermal cell in a hypotonic solution or water molecule which is that is the water um, molecule in the solution is greater than that of the cell volcule. So it is going to attract. That means that the cell volcule is going to attract more, take in more water, and when it takes in more water, like, it's going to make the cell become turgid. That is all that is trying to say, and that's all the meaning of turgid. So importance of turgidity. So turbo pressure or turgidity keeps the leaves and stems of herbaceous plants stiff and upright, providing mechanical support to the plant. It keeps the stomata of leaves open. It causes the leaves of insectivorous plants to capture insects. Yeah, as you know. And when a plant cell is placed in hypotonic solution, water molecules move out of the vacuole through cytoplasm, cell membrane and cell wall to the surrounding solution by osmosis. This is because the surrounding solution is highly concentrated than the solution of the cell sap. The cell becomes flaccid if the water loss continues. The protoplasm pulls away from the cell wall, leading to plasmolysis. Plasmolysis is the shrinking of the protoplasm away from the cell wall by osmosis when a plant cell is placed in a hypotonic solution. Yeah, so that is it. Can hear the explanation from it. Okay, so let's go on. And so this is another subtopic 2, and which is animal cell in hypotonic or hypotonic solution. And if an animal cell into bracket red blood cell is placed in hypotonic solution, water molecules move across the cell surface membrane into the cytoplasm of the cell by osmosis. The cell content being highly concentrated than the surrounding solution will cause the cell to swell. Okay. As water continues to move into the cell, pressure of the water causes it to burst as a result of the flexible cell membrane. This phenomenon is known as hemolysis. So yeah, this one, the hemolysis, talks about the animal cell. So I hope you understand. So and I think the turgidity talks about the plant cell. So if an animal cell into bracket red blood cell is placed in a hypertonic solution, the cell loses water by osmosis by osmosis across the surface membrane to the surrounding solution. This is because the solution around the cell is highly concentrated than that of the cell content. This will cause the cell to shrink and eventually wrinkle. This condition is known as crenation. So that is it. So that is it. So summary. In this lesson, yeah, we have looked at the importance of substances to move in and out of a living cell, mechanisms of movement of substances across the cell, into brackets, diffusion, osmosis, and plasmolysis. So, simple experiment to support the concept of diffusion and osmosis. So that is that. So thank you for watching the full video, and I hope you understand everything. So all this is just talking about is about like the movement of substances in and out of the cell, and the processes by which um, the cell goes through in order for that process to occur, for the cell to become healthy. Yeah, so that is that. So thank you. Please subscribe and like my channel. Thank you. Okay, bye.